All right, guys, we're on to the last part of chapter 8.4 on stem cells. So we're looking now at the applications of stem cells in therapy. So if we look at the VC study design, for the key knowledge descriptor here, we need to know about how stem cells can be used in medical therapies. So the learning goals are right here, and a reference in your textbook on pages 375 to 378. So stem cell therapy, it's probably useful to define what it is. So stem cell therapy, what is it? Now, stem cell therapy can be broadly defined as the generation, that is the production of healthy, laboratory-produced or laboratory-cultured tissues and organs from stem cells to replace damaged cells in the body. So this is the crux of stem cell therapy, that that stem cells can be used to produce healthy tissues and organs that we can then transplant into parts of the body where there are damaged cells in order to replace or help repair and regenerate those damaged cells. Now, stem cell therapy comes with its challenges, and for it to be successful, there are a few things that need to be achieved. So, for stem cell therapy to be successful, we need to achieve 4 things. The first is that we have to be able to ensure that stem cells can replicate themselves in cultures within the body. Sorry, within a laboratory, cultures within a laboratory. So often, if stem cells are taken out of the, let's say, an adult or an embryo, and then they're cultured here. So these cells need to be able to grow in culture. You have to be able to grow rapidly so that they can later be used in transplants. So the second of the challenges is that stem cells must be able to differentiate. Able to differentiate into the particular cells required to treat disease. To treat a certain disease or disorder. So if we're going to extract stem cells from a body and then get them to treat something, they must be able to differentiate into particular cells that we need to treat whatever problem a particular patient is having. So if we're going to have a stem cell, it needs to be able to turn into what we want it to turn into. Must be able to do what we want. And we have to be able to choose exactly which one of those specialized cells are differentiated into. So if we have issues selecting a particular cell type, from a stem cell, then that poses a significant challenge. Now, the third challenge is that stem cells must not remain as self-renewing stem cells. So that is, they cannot continue to renew themselves, and that is grow continuously, because if they grow out of control, we have a problem. So they must not remain as self-renewing cells with the potential to grow out of control. So once we have our cells and they've been grafted or transplanted into somebody, they must not turn into like a mass of cells. So we cannot have them turning into neoplasms or tumors. So that's the third challenge. Now the fourth challenge is that the immune system of the recipient must not reject the stem cells. Must not reject the stem cells or tissues created from stem cells. So these are the four challenges that must be met in order for stem cell therapy to be successful. Now, there are three types of stem cell therapies. So we need to know just what these are and their key advantages and disadvantages. So the first is adult stem cell therapy. This is just where you extract like adult stem cells from the body and then use them to transplant them back into a body to replace damaged cells. It's pretty simple. And a couple of examples are bone marrow transplantation, and this is to replace lost bone marrow stem cells, which are necessary for the regeneration of cells that can be found in the blood. Another one is epidermal, that is skin stem cell transplants. So this is a good one for where like significant amounts of skin have been lost, such as through third-degree burns. They need to be restored. Now, the key advantages of adult stem cell therapy, well, the key advantage, we'll just talk about one, is that it is the least problematic of all the stem cell therapies. So it's the least problematic, because each kind of adult stem cell is not extremely potent, so it can only differentiate into so many kinds of cells, like very few kinds of cells, which makes it the least problematic. Key disadvantages are there are very few key disadvantage, just looking at one, there are very few adult stem cells in the body. To be harvested. So it takes quite a while to get enough of them. So that's the first of the types of stem cell therapies. And the second one is embryonic stem cell therapy. So as we know, embryonic stem cells, they have an enormous potential. They're totipotent or pluripotent. And because they have high levels of potency, they can actually turn into any kind of tissue or organ that we want. theoretically. So their key advantage is that they can differentiate into any kind of cell. Into any kind of cell or tissue, theoretically. The key disadvantage is that the key disadvantage are there are a lot of ethical considerations and moral objections to the use of embryonic stem cells. The third type of stem cell therapy are the induced pluripotent stem cells, or the iPSCs. So the induced pluripotent stem cells are adult cells, somatic cells that have been genetically reprogrammed so that they become embryonic stem cell-like. So they're like adult stem cells that have the potency of embryonic stem cells, which makes them have the same key advantage as the embryonic stem cells. That is, they can differentiate theoretically into any kind of cell or tissue. But their key disadvantage is that they need to be reprogrammed, and that requires time and technology, specialized technology. Okay, so here are the three kinds of stem cell therapies: adult stem cell therapy, embryonic stem cell therapy, and induced pluripotent stem cell therapy. And each of them has its own answer to the various challenges. iPSCs, actually, I should add one more key advantage. Since usually with induced pluripotent stem cells, you would extract somatic cells from one person, induce them to become pluripotent, and then turn them into whatever tissue is required to restore or regenerate a damaged part of their body, there is no risk of rejection. No risk of rejection by the immune system of these new cells because they belong to the body. Usually rejection occurs because the body recognizes that those cells are not part of the body, that they're foreign matter, so they destroy them. Okay, so we need to be aware of the ethical implications in stem cell research. And there are some major ethical considerations for embryonic stem cell therapies because of the nature of embryonic stem cells, that these cells are often derived from embryos, each with the capacity to grow into a fully functional human being. And so some people consider the extraction of cells from embryos for these therapies as being like destructive to human life. Some people also object against the reprogramming of cells to make them induce pluripotent stem cells. The reprogramming of cells is like playing God and changing the genetics of certain cells to meet your ends, and that raises some moral objections as well. All right, so here we've looked at the therapeutic applications of stem cells, the different kinds of stem cell therapies, the challenges involved, and some of the ethical implications.